That's a good question, isn't it? Nope, not that one, guy.
Oh, is it this one?
Yeah, that one. Hang on here, I'll just get logged in. Yeah. Just the okay. If not, I can just go and do this. Okay.
It might be time getting the microphone to
Okay.
the speaker.
Sounds good. So questions at the end. Yeah.
Yeah,
Okay.
yeah.
And
That's all
this
good.
live.
You're
Yeah.
live.
Yeah. That's right. Just noticed. Okay. Thank you.
Okay, so you're all set?
Think so.
Okay, so 8 minutes, Craig, no problem. 8 minutes to talk?
Sure.
Welcome everybody to the first keynote address of CHARM 2017, and it's my absolute honour to introduce Associate Professor, or Assistant Professor, excuse me, Craig Jenne from the University of Calgary. Craig completed his PhD in the 2000s, I won't nail you down on the date, and then he was involved in a postdoctoral, in a collaborative format, which is actually quite unusual, funded by NIH out of University, California, San Francisco, and Australian National University. So he's actually a Canberran, not by birth, but by association.
Months, yeah.
So he's quite familiar and shocked with how downtown has changed in around the Civic Centre interface. He also is the Director of Science at the Snyder Centre for Translational Research. So he has lots of weekly meetings with doctors, sees patient samples, and hopefully today with some of that data will be incorporated into his talk. His publications, I'm amazed with Science, Nature Med, Nature Immunology, Invited Reviews, JX Med. So he's an outstanding investigator at the peak of his talents and at the peak of his discovery arc, I would say, and we're fortunate to have him here today. He did just arrive yesterday, so hopefully
fall asleep.
I don't know what time it is in Calgary. Anyway, we look forward to your talk, and without any further ado, hand over to you. Thanks.
Thank you very much. I'd just like to thank you for the invite to be here. Very excited to come down and show you some of the stuff we're doing and maybe present some data from a slightly different angle. And what we do is we study mice and we try to put it in the context of human clinical questions. And we use a rather unique technique, and I'll get into that in a couple slides, where we actually put the microscope into the live animal to do intravital imaging. And one of the questions that caught me during one of my postdoc projects was this link between clotting and infection. And that we know that the vast majority of patients with systemic infections, sepsis, but even some cancers have clotting problems. And this is often seen as, too much clotting, circulating microthrombi, things like that. We also know that loss of circulating platelets, thrombocytopenia, is associated with really bad outcomes in things like sepsis. So as platelet numbers drop, the probability of a good outcome of patients with sepsis also drops. And one thing that's related to this observation is this phenomenon known as disseminated intravascular coagulopathy. So this is, again, those generation of spontaneous small clots throughout the body. And this really is a twofold problem. One is we absolutely do not want circulating clots. They can cause embolisms, they can cause things like stroke or myocardial bleeding in these patients. And to give you an idea how big of an issue this is, just a quick PubMed search shows if we look for coagulation and infection, there's more than 10,000 papers out there. So there's a lot of information, a lot of people looking at it, and yet we're really not getting too far in coming up with a solution. Now, given the association between clotting and these things like sepsis or other infectious diseases, it's logical that we've tried a lot of things, and we have at our disposal a number of anticoagulants, and basically anything that's been approved has been tried in sepsis. So things like antithrombin, promising results early on as the clinical trials got bigger, more than 2,000 patients, we found that there was only a marginal improvement in survival. As we move to other proteins, such as activated protein C, when we get in, once again, to the larger studies, we saw no protection. Thrombomodulin, protection in cancer, none in sepsis. And the current one that's underway, at least in Canada, is a very large heparin study. Things are looking good so far, but it's not a silver bullet. And the question really becomes why? If clearly clotting is the problem and we add anticoagulants, why can't we help these patients? And I think part of it is that we're likely still targeting the wrong thing. And that is, clotting is likely the end result of a problem upstream. And if we're only targeting the end product, we're likely not solving the situation. And from our point of view is this concept, and I'll come back to this at the end, is that we need to uncouple clotting from infection. So again, there are times where coagulation is absolutely critical. We can't do surgery on somebody who's received maximal doses of anticoagulant. Likewise, patients coming in with trauma, bleeding has to stop first. So these patients are not really eligible for the high-dose anticoagulants. And this is likely not the problem. The problem is somewhere associated with the immune system and not specifically with hemostasis. And this is sort of the background of the project that we started. And the very basic question is, how is inflammation and coagulation linked? Now, to address this, we developed a couple, at least initially, very simple models. And I just want to lay out how we approach this as an immunologist. And basically, what we know about the immune system is that if we look at infection over time to resolution, there's really two phases. There's a very rapid innate immune response, neutrophils, monocytes, things like what's linked to coagulation. And the model we developed is really simple. We take our little patient here, and we simply inject it with bacteria IV and watch what happens. And when we do this, and we look for where the bacteria go, we check all the usual suspects, so spleen, kidney, lung. The bacteria really are not going there. So where do bacteria go when they're put into the bloodstream? In fact, they all go to the liver. So the liver is your principal immune organ when you're dealing with a systemic infection. Why is that? Well, a lot of it has to do with how the liver is actually designed. And if we look at the anatomy of the liver, and we look at the blood flow, we can see here that there's blood coming in through the hepatic artery, but also the portal vein. So what is the portal vein? Well, this is what's draining basically the gut. So any pathogens entering your body through the GI tract come in through the portal vein and then enter the liver. And the liver is designed as a honeycomb. And basically, this network of sinusoids slows the blood flow down, forces it in contact with the cells that are in the liver, so we can screen for pathogens. And if we look at my artistic interpretation of a liver, we have these columns of hepatocytes, so the principal cell in the liver. They're lined by a leaky endothelium. But the key feature of the liver is what lives inside the blood vessels, and that are the Kupffer cells. So these are professional macrophages. They live right in the bloodstream, and they're found throughout the liver. And it's these guys that detect the invaders, catch them, and try and kill them. So how do we understand what's going on? Well, older days, it was simply done on a tissue, such as a frog's tongue, that was thin enough to see through. So you somehow sedate the frog, or pin it down, or I'm not sure what the ethics were in 1839, but you would take the frog, stretch its tongue out over a microscope, and you'd watch what's happening. And if we look in Conhein's notes, we can actually see his sketches of immune cells here rolling along the walls of a blood vessel. So he had no idea really what these cells were, but he could make the initial description. Today we use a slightly more sophisticated system. So we use a resonance scanning confocal microscope. And basically the approach is still similar. We sedate the animal. We do a small surgery to expose, in this case, the liver and place the animal on the microscope. And then a series of lasers will excite dyes we put in, come back out through a digital filter, and we capture various fluorophores or colors being emitted by the labeled cells in the animal. And with this, we can directly track, for example, what happens to those bacteria we inject. So this is a picture of a mouse liver. The green are the columns of hepatocytes. The little black lines are the sinusoids, or the blood vessels through the liver. And these red cells are those Kupffer cells. So we labeled them with a fluorescent antibody, and now we can see the Kupffer cells. And what we're going to do is inject a fluorescent green labeled Staph aureus. And if we watch this time-lapse video, we can see how quickly these Kupffer cells grab the circulating bacteria, such that by the end of the video, which is about a 10-minute video, there's no circulating bacteria left. So the liver is extremely efficient at catching these bacteria. So once we've pulled them out of the blood, how do we get rid of them? What happens next with those bacteria? Well, again, this is typically an innate immune response, so we wait for the neutrophils to come in. They're going to eat things, they're going to kill the bacteria. So here's a healthy, untreated liver. We can see there's a couple neutrophils in here. And if we look in an infected or challenged liver, we see this really profound neutrophil recruitment. So this is 8 hours after viral challenge. We get a similar response 4 hours after bacterial challenge. But basically, all of the blood vessels of the liver fill up with neutrophils. If we look more closely, an untreated liver, the neutrophils are truly thrilling structures that just sit there and do absolutely nothing for the 4 hours you image them. If you look at an infected animal, though, we start to see this. And these neutrophils are actively searching for things. We can see this one here, for example, not actually moving, but continually sticking arms out, scanning for whatever called it to the liver. So there is this very dynamic process that occurs within the liver vasculature. If we look, it doesn't matter if it's viral, so double-stranded RNA is a stimulus, or a bacterial product, a lipopolysaccharide. They're both just as efficient at bringing neutrophils in. Now, this all makes sense. Bacteria, early response, innate immunity. But one small aspect is the time delay. So we're catching the bacteria, but then it takes about 4 hours for the neutrophils to get there. So what's happening in those first 4 hours? So you can imagine, again, if we look at this at high resolution, neutrophils are coming in somewhere over here, and there's this little bit of a gap. And if we think about what's happening in that gap, well, some of the bacteria are sticking to the Kupffer cells. Hopefully, they're going to get phagocytosed, internalized, and killed. But we know that over the course of 4 hours, we don't get them all. So some of these are going to stay on the surface of the bacteria and of the Kupffer cell. And within 4 hours, they replicate for multiple generations. So we're not basically eating them fast enough. And that if left unchecked, we're still going to succumb to the infection. And what this means is that you're going to kill the Kupffer cells. And more importantly, if you're the mouse, doesn't work out well. So there's something else in there. There has to be something that we don't need 4 hours to bring in. And it turns out the answer to that is found in these guys. So the earliest form of really innate immunity we have are these things called hemocytes or amoebocytes, which are really half platelet, half neutrophil in the same cell. So platelets, innate immunity, it doesn't really instantly come to mind, but when we look at a platelet, they're in fact the perfect immune cell. They're found constitutively in the blood in insanely high numbers, billions per milliliter. So they're always there. You don't have to recruit them. They actually express immune receptors, TLRs and FC receptors. So they're primed to detect these pathogens. And when they activate, then they can control the immune system. They modulate the function of other immune cells. So we asked, well, maybe platelets play a role. So here's that same experiment. We're going to inject bacteria. This time it's a bacillus instead of Staph aureus. Here, instead of the Kupffer cells being in red, the platelets are in red. And we'll inject the green bacteria. And if we watch what happens here, lots of platelets circulating. But as the liver catches these bacteria, platelets start to accumulate on them. So within seconds, the platelets have identified the bacteria and begin to respond to them. If we look sort of more closely, we can again see in a control animal, individual platelets freely circulating. In the animal injected with bacillus, where it catches a bacteria, we get platelet aggregates forming around the bacterium. If we test this in vitro and simply add bacteria to platelets, once again, we can see platelets now binding the bacteria, platelets aggregating with each other, making large granular type complexes. So we wondered, well, this must be playing some kind of role. So we looked a little closer, and the advantage of confocal microscopy is we're not limited to the one focal plane I've been showing you. In fact, we can take a series of focal planes, take those individual pictures, and then reassemble them back into a 3D model. And when we do that, we get these really interesting pictures of some bacteria, so in this case, methicillin-susceptible Staph aureus, binding to Kupffer cells, nothing else happening, the animals survive just fine. If we use the nastier drug-resistant Staph aureus, we get these platelet domes that appear to form and seal off the bacteria. And once again, if we look at this over time, so these are 3D images taken every minute, we can see that this Kupffer cell catches a bacterium, and over the course of about 1 minute, the platelets begin to aggregate such that by 4 minutes after capture, that platelet is completely, or the bacterium is completely encased in platelets. We thought that this is rather interesting, and that this must have something to do with immunity. So we got rid of the platelets. We said, if this is important, then if we get rid of the platelets, something bad will probably happen to the mouse. And in fact, we were right. If we look at, again, that nasty bug, the drug-resistant staph, and we get rid of platelets, the mice now no longer survive, and that they need this immediate platelet response to fight off the infection. So although neutrophils are a key cell later, if you don't have platelets in that first window, you lose the battle to the bacterium. What was happening? This is still open for debate in the lab, but we found a number of mechanisms. So for example, if we injected bacillus with platelets that are unable to aggregate, so GP1B, which is a platelet adhesion molecule deficient animal, we saw increased liver damage. So the hepatocytes were dying. We also saw an increase in hematocrit, and we measured the hemoglobin, and it wasn't going up, so it suggested the liquid fraction of the blood was getting out of the blood vessels, leaving the red cells behind, so we had leaky vessels. And most importantly, if we injected propydium iodide, this red dye, we could actually see dead and damaged endothelial cells and Kupffer cells. So without those platelet domes, the bacteria were secreting toxins that directly killed hepatocytes and Kupffer cells, as well as causing vascular leakage and other problems throughout the animal. So very reminiscent of a sepsis, severe sepsis phenotype. So platelets were that first line of defense that protected us from all of this. So just to put this into a bit of a model, under basal conditions, most platelets are free-flowing. We did see some, however, were interacting with the Kupffer cell. They would come in for a second or so, and if there was no bacteria, they would leave. So there was this active immune surveillance happening, and this was mediated by GP1B on the platelet, and in fact, von Willebrand factor that was in the blood and was being pulled out of the blood by the Kupffer cells for disposal creates this little landing pad for the platelet. If we added a bacteria, depending on the one we used, platelets would identify this, would bind to it, and immediately switch this touch-and-go screening to this long-term adhesion and basically aggregation until we sealed that bacteria off from the rest of the body. If we got in and interfered with some of these molecules to prevent the adhesion, then we could still see the occasional platelet interact, but we weren't able to form those domes, and what happened was death to hepatocytes and endothelium, as well as significant damage to the Kupffer cells themselves, allowing the infection to win. So just as a summary, we really think that this is an example of what we can do with this intravital microscope, in that without seeing these dynamic interactions, we probably would have never realized that platelets responded immediately to the bacteria, but even before the infection, we're actively screening and looking for it. So from there, we expanded. And we said, that's what's happening in the first 5 minutes to the first hour. What happens later? What else are platelets doing? And if we look again in the liver of an uninfected animal, we see these free-flowing individual platelets. Some are touching down on these neutrophils, but there's no large aggregates or anything in here. If we challenge this mouse with a virus or a virus mimic, and we look 8 hours later, we get this. So now we can see these really clear platelet aggregates, and they're occurring throughout the liver vasculature. This is why these animals become thrombocytopenic. So the platelets are no longer circulating. They're stuck in the lung, they're stuck in the liver. We can reproduce the clinical thrombocytopenia simply by injecting double-stranded RNA. And if we block the adhesion molecules, we prevent thrombocytopenia. We can go in and measure the size of those platelet aggregates. So take some pictures, measure how big each one of those little platelet clumps are. And we can find that, following, again, viral challenge, we see a dramatic increase in small aggregates, but even aggregates out here that are bigger than a leukocyte. So these are large collections of platelets. And depending on which adhesion molecule we block, so CD41 on the platelet or CD11B on the neutrophil, we can basically completely attenuate this aggregation process. And I'm going to come back to the CD41 in a couple slides. So we've been able to map this using the microscope. If we look a little more closely, again, a really boring, healthy liver, not much is happening with the platelets. If we give this mouse poly-IC, we get this really interesting phenotype. So those aggregates we see are not clots per se. They're not stable. They continually form, and you can actually see platelets that appear to roll along the surface of the neutrophil, form an aggregate downstream, and eventually rip off and re-enter circulation. So this is a very dynamic process. We can do 3D imaging of this, again, looking at a control liver, individual platelets. If we look at a virus-infected liver, so this is with myxoma virus, and the infected cells turn green, we can see these really large platelet aggregates, many of which appear to be associated with the neutrophil. We can pull individual cells out and take a look. So again, control maybe 1 platelet per neutrophil. When you give the viral challenge, we get aggregates the same size of a neutrophil. So this is a very active process. And we can also take these 3D pictures over time and reanimate them back into what we would term a 4D movie. And this gives us a really nice idea of what's happening. So if we watch this cell up here, we can see platelets binding upstream, rolling across the side of the neutrophil and forming this aggregate that then dissolves and falls apart, re-entering circulation as individual platelets. So if we were to take a simple histology slice of this liver, we'd probably misinterpret this as coagulation, whereas in reality at this time point, it's actually neutrophil platelet interactions and likely doing something else other than creating thrombi. So that got us asking, so if it's so dynamic, what exactly is the point of this? What are the platelets accomplishing? And one thing we've had seen in previous studies was that when it comes to intravascular or systemic infection, platelets are a key player in the generation of these nets that we saw a couple talks ago, so these neutrophil extracellular traps. So if we look in a systemic infection with E. coli and we use an isotype control antibody, we can see that in untreated animals, we have no nets. If we give LPS from E. coli, we get lots of nets. And then if we get rid of the platelets, we get rid of the nets. So in a bacterial infection, platelets being present leads to the formation of these nets. For those that are not familiar with these neutrophil extracellular traps, what are they? Well, basically, if you stimulate neutrophils under the right conditions, instead of phagocytosing or degranulating, they actually undergo another mechanism of immune function where they take their nucleus, decondense all the DNA, and eject it out of the cell. So basically, they spit their nuclear DNA out and spread it in the microenvironment as a spider's web or a sticky net. And what happens is pathogens get caught in this. Now, the net is not just DNA. As they're doing that, it's covered in all the DNA proteins that would normally be there, like histones. But they also release their granular proteins, so their elastase and their proteases and their MPO and everything else is found on these nets. So it's a very cytotoxic structure. And basically, any bacteria or virus that bind to these nets, there's a significant number of them that die. So this is an effective immune strategy to kill bacteria. Now the question is, why do we need that? We've always known neutrophils to be sort of the front line defense to bacteria. They eat things, they have oxidative burst. And part of the answer can be found in the immunology Bible known as YouTube. And that is, how good are neutrophils at eating bacteria? So we have a neutrophil here, and we have a single bacterium over here. And we'll start this video, hopefully, and we'll watch this neutrophil chase. He's going to ignore this one. Come back for that one later, and don't worry about that one yet. Still going. Another bacteria in here. As you can sort of imagine, if this was the efficiency of your immune system, we would all still be sea urchins, right? Like, we're not good enough to chase down bacteria. So if we're not fast enough to catch them, how else do we do it? Well, we're going to spread that trap and let the bacteria come to us. And just to prove to you that this isn't something that's, mouse specific or only found in a petri dish, part of the opportunity / pleasure of my job is I get to go and help drain human abscesses and bring back to the lab little tubes of really nasty stuff. But if we take those and we look at them, so an abscess is often filled with infiltrating neutrophils. We can stain for neutrophils in red here and total leukocytes in blue. And we can also add a stain for DNA. And when we overlay these, we see these really healthy trilobe neutrophils. But we also see this neutrophil here that has 3 empty spots where it used to have a nucleus, and surrounded is this diffuse sticky web of DNA. So we can show that these nets actually happen in human samples. Do they work? Well, if we look in the mouse, because my grad student won't go under the microscope, we can see a neutrophil here that's released its DNA into the blood vessel of the liver, and within that DNA, we've managed to catch 2 E. coli. So these nets are a very effective strategy. If we look in a virus infection, we get the same phenomenon. So looking at an untreated animal at the top and a myxoma virus infected animal at the bottom, we can stain for components of nets. So histone, which is normally a nuclear protein, if we add the antibody to it and the cells are intact, we should get 0 labeling. So the only stuff we're seeing is stuff that's outside the cell. And then we add elastase, which stains a granular protein. The only place these two should be in the same spot is a net, and we get this beautiful co-localization throughout the vasculature of the liver. So we can demonstrate whether it's bacterial or viral, human or mouse, nets are a very common immune effector mechanism. And finally, to prove to you that the platelets are the key player here, we have our untreated mice and we're measuring elastase. We do poly-IC, double-stranded RNA, we get a nice response. Give a live viral infection, we get an even better response. And if we block the platelet's ability to stick to the neutrophil, we completely block the nets. So the neutrophils are still there, the virus is still there, all the cytokines are still there. They simply can't aggregate. And when you do that, we don't get nets. Now, as with everything else with immunity, this doesn't come for free. So all aspects of immunity are sort of a cost-benefit ratio. So you can imagine if nets can kill bacteria and viruses and parasites and everything else, they probably can kill us too. So the body is making these structures in a sort of last-ditch effort to stop the infection, knowing that there's going to be some self-damage. So nets have been shown in cell culture to kill endothelial cells, something we probably don't want happening in our bodies. Individual components of the nets, if you pull out all those individual proteins, they can actually activate thrombin, so initiate clotting in vitro. And that led us to ask the question about, are nets, is the way they damage our tissue, is it the way they kill the endothelium because they're turning on coagulation? And can this coagulation, if it's happening, actually damage the organs, or is it just a small byproduct of the net? So one of the first things we did is we looked for common markers of clotting. So Pi1 and thrombin-antithrombin complexes, and we find that if we inject our bacteria and we pull the blood out of the mouse, we can detect markers of coagulation. So we wanted to use that and say, okay, we believe clotting's happening. How are we going to see clotting? How are we going to use our microscope to visualize an activity as opposed to a solid cellular entity? And what we developed was a protein that not only detects the presence of thrombin, but detects only active thrombin. So prothrombin's in our blood at all times. When it's activated, it's quickly inactivated, becomes thrombin-antithrombin complexes. So we wanted to know, is there active thrombin in a tissue? And what we've been able to optimize is this kind of reporter approach. So in this protein, we have a fluorescent dye that's glowing all the time. At least on the microscope, it's glowing all the time. The other end of the protein, though, is this little tail that actually absorbs all the light that's coming from that fluorescent protein. So when we look at this intact protein, we see no fluorescence. The piece that connects the two is a very specific peptide sequence. And in this model, the peptide sequence is exactly what's recognized by thrombin. So if thrombin is present and active, it's going to cut that linker, release the quencher, and basically will reveal the fluorescence that's always on that protein. Does it work in mice? Oh, here's our infected mouse liver, lots of neutrophils. And what we'll do is start the video and we're going to inject this thrombin dye. And you'll quickly see what happens here. So there's actually active thrombin throughout the liver during the infection, and consistently active. Whether we look at 4 hours or 8 hours, we're picking up the same degree of thrombin staining. So thrombin is consistently being activated within these sites of infection. And if we take still images, we get this really beautiful picture of how big of a problem it is. So most liver sinusoids have thrombin activity. If we look a little more closely, we can find that the areas of the liver that have the highest thrombin activity, so again, challenged with LPS, are the ones with these little red specks. These are our nets. So where these neutrophils are releasing their DNA and making nets is where we're turning on thrombin. And in fact, if we treat the mice with an enzyme to break down the nets, so DNase, or we use a mouse, such as a PAD4 deficient mouse that cannot release nets, neutrophils still activate the same way, but they simply can't release the net, we completely attenuate the thrombin signal. So we know that this isn't some byproduct of systemic inflammation. It's not cytokine-driven. It's not monocyte-driven, not tissue factor-driven. This is driven directly by the nets. If we look again, do a different stain, this time we wanted to show that it's not just thrombin that's activated, but thrombin's doing something. So we look for the deposition of fibrin, the key component of a clot, and we can see exactly the same staining pattern. So these liver sinusoids are now filling up with fibrin deposition. And in fact, if we stain the fibrin in the nets, we get a near perfect overlay. So wherever there's a net, we're finding fibrin. And this is partly due to the fact that there's some hints in the literature that nets also prevent the breakdown of fibrin once it's produced. So nets are driving thrombin and also preventing fibrinolysis. What's the overall impact? Well, the nice thing is, because these are live animals and real-time videos, is we can actually inject contrast agents and look at blood flow through these tissues. And what we find is if we look in the liver and we inject a contrast agent in blue, these vessels that have thrombin activity don't light up blue. So we can actually count the number of non-perfused blood vessels in the liver. And if we were to treat this animal about an hour before we do the imaging with the DNase to break down any nets, which both will stop thrombin activity but also allow fibrinolysis to happen, we actually open up all those blood vessels again. So the presence of nets not only triggers thrombin, but actually causes perfusion problems through the microvasculature. And if we count that, we can see, once again, an LPS challenge, we have lots of occluded vessels, and if we either break down the nets with DNase, we had a PAD4 deficient mouse, cannot make nets, or even directly inhibit thrombin, we open those blood vessels back up again. So the recruitment of neutrophils, the recruitment of platelets, that's not what's causing the ischemia. It's actually nets activating thrombin. And then finally, is this damaging the organ? Well, if we look at a marker of liver damage, ALT, again, infection, lots of ALT. If we prevent nets, we attenuate that response. Or if we add DNase, starting to trend in the right direction. It's not as effective as stopping the nets entirely, but we can open some vessels back up. So at this point, I'm just going to show a bit of a model, and then I want to show you our latest stuff, which, if everyone in the lab is still working while I'm here, should be submitted this week. If not, then they'll wait for me to get home and get mad, and then we'll submit it. But it's going out soon. So the model so far, we're trying to build off this idea of why do we recruit neutrophils, and specifically what are platelets doing. And if we look in the liver, systemic infection with anything, bacteria, virus, even analogs, we get this really profound and rapid neutrophil recruitment. They seem to form a landing pad for platelets, and that once this initial platelet docks, then we get this GP2B3A interaction, which allows platelets to aggregate. The end result of this is really a signal back to this neutrophil to get it to release its nucleus and create this net. And this is where things get a little bit more interesting. This net is actually highly pro-coagulant. So there's a lot of things on the net that interact directly with coagulation. So neutrophil proteases, histones, polyphosphate on the surface of a platelet, and platelets can also bind microparticles with tissue factor. All of these support the generation of thrombin. And once thrombin's activated, we get the formation of a small clot. Now, if this is where everything stopped, we could probably mitigate the damage. But again, as pointed out in an earlier talk today, these clots tend to feed back on the platelet. And we get additional platelet recruitment to sites of thrombosis, additional platelet activation, which then results in an even bigger thrombi. And we get this vicious amplification circle, which eventually leads to poor perfusion through organ microvasculature. Everything I've shown you was the liver, and we wanted to see if this was, again, a liver-specific phenomenon, or could we extend it to something else? And at the time we were doing this, the liver is actually fairly easy to image. It's a big organ. It doesn't beat like the heart, doesn't breathe like the lungs, so you can sort of tease it out of the animal and place it on the microscope and image it quite well. We wanted to get another model up and running, so a postdoc in the lab got a lung model up and running. And this is a little trickier because as you're trying to take pictures, the mouse wants to breathe. It sounds silly, but we've come up with a way to stop the mouse from breathing and keep it alive. And we actually have a little vacuum that holds the lung against the microscope. And the microscope is programmed to a ventilator so that every time the mouse breathes, we inflate the lungs, we take a picture at maximum inflation, and the microscope is timed with every breath. And with that, we can now generate these really beautiful images in a live animal. So these little black spots are the air spaces, and then we get a couple different leukocyte populations. So we started and basically retraced our steps that we did in the liver, and we used a virus infection. So happens that one of the ones that works well in mice is H1N1 flu, so that was the model we chose. And what we found is in a resting lung, there's actually a lot of neutrophils that live there full time. But if you infect with flu or use a double-stranded RNA mimic, we can recruit neutrophils quite rapidly to the lung, and we could see these with a microscope and count them. We could also find these cells getting into the airspace. So if we lavage the lung, we would find cells in the lung. And we could also do a myeloperoxidase activity, so an in vitro assay that basically approximates the number of neutrophils in a tissue. And whether it was live H1N1 or poly-IC, we got pretty much the same response. We also have a difficult time measuring lung function in mice because the machines are really expensive. So we've come up with cheap ways to try and measure how much damage there is in the lung. And one of them is actually to measure the weight of the lung when you remove it from the animal, and then you put it in an oven for a couple days and dry it out and weigh it again. And this gives us an estimate of how much fluid was actually in that tissue. So a marker of pneumonia, for example. And again, whether it's live influenza or poly-IC, we can definitely see increased fluid in the lungs following challenge. Now, what happens to the platelets? again, if we look in a control animal, there's lots of neutrophils that are here under baseline conditions. But if we start the video, you're going to see that all these platelets are sort of individual little dots. We can still see some flowing through the larger vessels in here, but there's no real aggregation, and there's very little platelets interacting with the neutrophil. If we look following poly-IC, just like the liver, we get this really profound phenotype of platelets aggregating and adhering within the vessel, but also adhering directly to the neutrophils. And just a different field of view, again, we can see these huge aggregates all in the neutrophils, basically occluding these vessels. Larger vessel here, wide open, small little capillaries around the airspace completely obstructed. So we go in and we measure this using exactly the same approach we had in the liver. So measuring the number of aggregates that are 10 square microns big, 25, 50. So again, the size of a cell, we're getting large platelet aggregates. And where are these platelets? Well, by staining the neutrophil and the platelet together, we can actually see that the vast majority of these green platelets are associated with a neutrophil. So it's the neutrophil that's providing that docking station for platelet recruitment. So far everything's lining up quite nicely with the liver. So we wanted to ask the next question was neutrophil platelet interactions, does this lead to NETs? So using histone as our marker, again, a control animal, lots of neutrophils, but no NETs. We do H1N1 infection, we get more neutrophils, and now we can clearly see extracellular histone, so that there is the presence of NETs in these animals. And then sort of the final question was, well, NETs before led to thrombin, so is that the case in the lung? Again, this is 0 minutes, we inject the probe, wait 1 minute, green is thrombin activity, so we can see these vessels are completely filled with active thrombin, and we can quantify that. So we wanted to understand, we thought this was a good opportunity to get in there and try and break that amplification loop. So if thrombin is bad, then we should see obstructed vessels. And here we can see a large vessel, free blood flow, completely obstructed parts of the lung here where you're not getting any blood moving through those capillaries. So poor gas exchange, poor organ function. So we said, can we get in there and break this apart? Can we therapeutically treat these animals, perhaps without giving them high doses of anticoagulant? So if the model is true in the lung as it was in the liver, we have neutrophils that are recruited. These allow platelets to dock, form aggregates. This will trigger NET release. NETs will generate active thrombin, and then we'll get fibrin deposition and clots. And as we saw in the liver, there's a strong potential for this thrombin to feed back on the platelets, and this is going to create an amplification. And this is what we want to get in there and stop. So can we stop this? Can we let the initial response to the infection go to control the infection, but get rid of this out of control amplification? And to do that, we took two approaches. One was directly block thrombin, which clinically we don't necessarily want to do. The other one is we hypothesized that thrombin was activating the platelets through their protease-activated receptors, or PARs. And the nice thing in mice is that unlike humans, there's the key PAR on platelets is really only found on platelets. So we can specifically target the platelet without inhibiting any other PARs in the mouse. So we're going to use our gatuban to block thrombin, and we're going to use a PAR4 antagonist to stop the signaling at the platelet. And what we found was really encouraging results, and that was when we did a flu infection, either use the thrombin inhibitor or the PAR4 antagonist, we blocked platelet aggregation. So we clearly stopped that feedback and additional platelet recruitment. And if we measure platelet aggregates, again, the PAR4 inhibitor, we reduce those significantly in blue, and if we use the argatroban, it's even better. When we looked for NETs, so platelet aggregates are down, we'd predict NETs should also be down. Control animal, poly-IC, lots of NETs, poly-IC plus PAR4 inhibitor, there's a few, but much less NETs. And if we quantify that, we can see that it actually brings it down to the same level as treating the mice with DNAs. So this PAR4 inhibitor was very good at blocking NETs. So we're quite happy with these overall results until we started looking elsewhere. And that was, if we looked at platelet recruitment in general, it's almost entirely blocked. But we also noticed that compared to a flu infection, there was fewer neutrophils coming into the lung. This creates a bit of a problem for our model, and that is, if the model was to stop amplification and further activation of platelets, how are we stopping the initial neutrophils from even being recruited to the lung? Which suggests that the platelets, in fact, are actually the initiator of lung inflammation. They're not simply responding to neutrophils. Somehow the virus and the endothelium in the lung are directly responsible for activating platelets, and it's the platelets that then bring the neutrophils in. So we had sort of our model backwards, and in fact, now we can target the platelet and completely inhibit all lung inflammation. And we can go ahead and measure these things. So for example, our wet-dry weight, we see less fluid in the lung if we use the PAR4 inhibitor. Total cells in the airspace are significantly reduced. And finally, MPO also reduced. And this is without targeting the neutrophil. So by blocking platelet activation, we reduce neutrophil recruitment and fluid leakage into the airspace. So just as a summary of that, just like other tissues, this general innate immune response creates these platelet neutrophil aggregates. They lead to NET release. NET turns on clotting, and things get very bad for the animal after that. We propose that there was positive feedback through PAR, and to some extent there is, but it's also blocking neutrophil recruitment initially and the initial platelet aggregation. So it's not just a feedback loop. The platelets are actually playing a role in initiating lung inflammation. So with this, we're again back to this point, and we think we have a reasonable path forward of targeting these protease-activated receptors to uncouple the infection-induced coagulation from hemostasis so that we can block some of these pathogenic pathways while letting the patient clot naturally to surgical or trauma challenge. And with that, I'm just going to thank the people that have made this all possible, including former postdoc supervisor, Dr. Paul Koobs, an intensive care unit fellow, Braden McDonald, who came in and did a lot of the liver clotting, along with Rochelle, and then Suk, who's done all of our lung work and basically got in that entire imaging program up single-handedly. And then a number of our collaborators over the years that have supplied us with platelets and also developed some of these early bacterial models, so Connie and Ella. And of course, the people that keep the lights on and the antibodies in the fridge. So with that, I'm happy to take any questions.
Obviously, Movember.
Just random.
Yeah. As it is. Thank you very much, Craig. Tour de force. Open for questions. We have roving mics for the live feed. So there's a couple of people that will come to you. So if we have any questions from the floor.
Thanks. That was a great talk. In your opening slides, you talked about the 4-hour gap between the initiation of, or the injection, and the arrival of neutrophils. I don't get a feeling of what's happening in that 4-hour gap. And the second thing is, what's the role of the Kupffer cell in all of this? You're focused on the neutrophil,
right?
But obviously the Kupffer cell is sitting there doing something as well.
Yeah. So two excellent questions. I'll maybe start with the second one first. The Kupffer cell will actively phagocytose and kill some of the bacteria, but can't deal with them all. Especially with things like the MRSA. It seems to proliferate too fast for the Kupffer cell. And in fact, some of the MRSA strains we tested actually live happily within the phagosome and start replicating within the Kupffer cells. They actually hide from the immune system until they've replicated enough. The Kupffer cell ruptures and releases them back in. Tied to that, though, is the role the Kupffer cell plays in initiating the response. So in the case of Staph aureus or E. coli, the neutrophil can see it just fine. The E. coli has TLR4, for example, for E. coli or LPS. In the other models, our viral models, poly-IC, for example, a mouse neutrophil is completely blind to poly-IC. It can't find it at all. And what we've realized is it's actually, in those first 4 hours, it's the Kupffer cell that's producing cytokines and chemokines and recruiting the neutrophils in. So in the case of bacteria, the neutrophils see it, but they don't know where to go. So in the first 4 hours is the Kupffer cell producing the chemokines, adhesion molecules, and cytokines to recruit the cells to that site. So that explains our small delay in there, is that the neutrophils know there's a bacteria, but don't know where it is. They need the Kupffer cell to upregulate adhesion molecules and chemokines.
In that viral model, if I can just sneak one in before the next question. So have you tried any of the neuraminidase inhibitors?
For the flu, no, we've not. We've started moving into instead of that, a vaccine strategy of what happens if these guys are aware of what flu is first. We get the same response on a recall, but we haven't got into the neuraminidase or the hemagglutinin either, trying to block adhesion so that we could get a viral signal from the alveolar macrophage, but not true infection. We haven't separated that yet.
Phil?
2 questions, if I may. Thanks, that was a fantastic talk. The first one, in the lung, you've tried the viral infection model.
Yeah.
Is in the liver, you did most of the work on the bacterial infection. Did you try the bacterial infection in the lung?
We did. So we did LPS and Staph aureus. They both nearly carbon copy of what we saw with the poly-IC. The timeline was a little different. So the poly-IC comes up after about 3 days, so you have to give 2 doses over 3 days, whereas the LPS, a single dose, you get a pretty robust lung response. But it's still boiled down to neutrophils, platelet aggregates, thrombin, and that if we inhibited that, we actually stopped the initial neutrophil recruitment. So we think that in the lung, there is this very clear, we believe it's tissue factor, but something from the endothelium platelet that results in the initial neutrophil recruitment, both to bacterial and viral infection.
Thank you. So the response is the same. The other question I had was just noting with the in ViVo model, you sort of found that platelets were protective. In vitro, you found the nets actually damaged the endothelial cells, and you need the platelets to develop the nets.
Yeah.
And there seems to be a bit of confusion there.
Yeah, I think this is, again, the idea of, our immune system in general has evolved to deal with these pathogens in the absence, for example, of modern medicine, right? So, they're all protective. It's just if things are left uncontrolled, the damage outweighs the benefit kind of thing. So if you could deal with the infection in the first few hours, you're never going to get to that robust net response. It's only when we really hit the mice with a lethal dose, for example, of flu or high dose IV of bacteria. In reality, you're probably never going to get 1 × 10⁸ cells intravenously unless you have a really nasty catheter infection. So we're pushing it to the limit to understand the interplay, but I think in reality, the few bugs you get leaking through the gut or something like that will be dealt with the Kupffer cells and platelets without getting to that really over-the-top response.
Yeah, up the back. So Craig, is the way we manage coagulopathy going to become troublesome? We're shifting from warfarin, heparin, through to these direct oral anticoagulants. Is that going to impact? So another way, you've talked about a trial where the people are studying heparin in this type of setting. So the old drug will turn out to be better than?
Better, I don't know. Yeah. I think we have to be careful of unintended consequences. So, for example, we know thrombin plays a role in this whole immune initiation through not just the PARs and the platelets, but endothelium, everything else. So things like thrombin inhibitors may have side effects we don't want. Heparin is a really interesting one in that it seems to be working, but I will guarantee if you ask 5 different people how it's working, you'll get 5 different answers, absolutely. Right? So, it's a direct anticoagulant, but it also neutralizes histones, it also neutralizes extracellular DNA, it neutralizes HMGB1. So, how it's working, I think is going to take another bunch of years to figure it out. But I do think we have to be very careful with these strategies of not just looking at clotting times and understand exactly what's happening too. So we're starting to get a little bit of evidence that actually suggests that interfering with these platelets might modulate the ensuing adaptive response as well. So again, we have to be pretty careful with what we're doing.
Yep, fair. Yep, in the middle.
So do you think this mechanism has an influence on priming adaptive immune responses?
Well, our initial results suggest that there may be a delay, for example, in the flu model, a delay in CD8 recruitment to the lungs. So, and that's simply using the PAR4 inhibitor. So we do think that, for example, platelet activation plays a key role in the generation of the ensuing adaptive response. And that's why we're now into these second dose type experiments. So give a lower dose, a non-lethal flu infection with and without platelet inhibition, and then come back with a lethal dose 8 weeks later and see if we did mess up the memory response on the first infection.
I'm never going to get those pictures of the liver out of my brain. That absolutely superb talk. I've been looking at people with sepsis for many years, and I've looked at their liver function tests, and it's been unclear to me what the mechanism has been. So that's very interesting for me to have that visual representation. It's a very naive question, the role of aspirin in platelet aggregation.
That's a very good question. That's aim 3 of my heart and stroke grant. We haven't started yet, but we wanted to know about even the auto feedback from platelet back onto itself, so ADP and everything. We haven't quite got there yet, but we did want to follow that out. One thing that we did pull from the records, for example, in Calgary, we don't have individual patient numbers, we have just global population numbers. And that is the rise in specifically the MRSA strains we're using that bind to platelets has paralleled the prophylactic use of aspirin in clinics. So the question is, was this a pathogen we dealt with quite easily before, and now that we've inhibited platelet function, we've provided a toehold for this infection to now sustain in the community? We don't have patient A was on aspirin and had an MRSA infection. We just have the population numbers from the Calgary Health Region, and they directly parallel each other.
Yeah, I wonder whether that's strain related, because you made the comment that here's the antibiotic sensitive staph, as though that was an effete little organism, but in fact they're highly virulent. And there's more variation between strains.
It's definitely strain specific, and even species specific.
Yeah.
So E. coli won't do this. Staph and bacillus do. So you're absolutely right. There's something about that specific bacteria that drives it, and not all bacteria do it. And it just, again, the coincidence that the strain we use in our mouse model is a clinical derived strain from Calgary, and that's the one that's risen at the same rate as aspirin and also some of the other Plavix kind of platelet inhibitors. They've risen in frequency together. So we're curious as to whether there is a mechanistic link or not.
And may I ask one more question?
Sure.
And that's that there's clinically
but at least in our model, it's pretty minor. We're not inducing liver failure, for example, but we've only really tested the influenza with that. So if we went with a lung bacterial infection, maybe. The one thing with influenza, at least in mice, is unless you give an insanely large dose, you cannot get influenza to the liver, right? So everything will just be, so it can't be antigen, it can't be antibody, it can't be, it's all within the lung. But something like a bacteria, once you break the epithelial barrier, will have access to other tissues, and it might spread. But what we've seen, it's pretty minor. The mice are sick because of their lungs, not because of something else.
Charmaine?
I don't know whether, yes, this is on. So you very nicely described the platelet neutrophil aggregates in your tissue or organ, a specific organ of interest. Is that also reflected somewhat similar to your previous question, distally, like in peripheral blood? Yeah,
so if we infect an animal with staph and we take peripheral blood and do flow cytometry, we see platelet leukocyte interaction, so CD41 positive neutrophils by FACS. We can also see, they're hard to spot, but they are in the videos, the occasional neutrophil circulating with a little platelet tag on it. So the platelets have stuck to the neutrophil while it's still circulating. So this definitely still has an effect. What we had found out from cell culture, though, is typically if those neutrophils are not adherent, they don't release nets. So the platelets will interact with the neutrophil, but unless you're getting that neutrophil stuck to something, it tends not to release the DNA. So you'll get the interaction, but you don't probably get those other pathological problems coming out of it.
Okay. I think that will draw this session to a close. I have just a small thank you gift, Craig, for the carry-on luggage.
That's right.
Thank you very much.
Thanks so much. Thank you.
Back at 2 for the 3-minute thesis, I think. Thanks, everyone.
Thank you. That was... Turn that off so I'm not live.